Well, thank you, Eric. It really is always a pleasure to be back here in San Diego, and I'm really pleased to be able to talk at this conference. And, it's kind of a hard talk to put together in 20 minutes, and, there's so much going on in our lab with looking at mechanisms of resistance to targeted therapies and looking at combination therapies. But what I wanted to talk about today was more of the efforts we're doing in a genomically guided trial. I think that will play nicely with what you'll hear from Laura, and also talk a little bit about where I think the future of cancer therapy needs to go, and I think that also dovetailed really nicely with what I think Roselle will talk about. I want to just give you some background. I got into this field of targeted or precision medicine for cancer therapy before translational research, precision medicine were even words that were invented, and started with the development of targeted therapy for chronic myeloid leukemia with the drug Imatinib or Gleevec. And the precept behind this was we knew exactly what drove the growth of these leukemia cells, an oncogene called BCR-ABL, an activated tyrosine kinase. We worked with a drug company to develop a drug to specifically inhibit this oncogene. And when we went to clinical trials, we saw that we took a disease which had a 3 to 5 year life expectancy, and now the CML expected life expectancy is 95% at 5 years, with a once-a-day pill that people take with modest side effects. So completely transformed a disease by targeting the precise molecular mechanism that drove its growth. Now, I've been giving talks for many, many years, and I've talked a lot about how do you translate the success of this to other cancers. And you have to start by understanding other disease. You have to understand the molecular pathogenesis to understand the appropriate disease targets. And people get that. That's precision medicine. The second precept is treat early in the course of the disease. And I think that's a precept that people haven't quite come around to, but I would continue to make the pitch that we have to treat early in the course, and I'll give you some examples of that. And then third, it is about matching the right patient with the right drug. So again, a critical precept of targeted therapy. So having that background, one of the things that one of my colleagues was giving a talk once at a major at ASCO, and he said, well, Druker had it easy. He worked on his dumb cancer. And I didn't take that personally. I was fine with that. But what he was really saying is he worked on a cancer where everybody was driven by the same molecular abnormality. And that's not the case with most other advanced cancers. So as I began to think and reflect on that, a lot of my own laboratory work has begun to work on a much more complicated cancer. And that's acute myeloid leukemia. The most common adult leukemia, 20,000 cases per year in the United States, and the median age of diagnosis is 67. So just remember that. Most patients are over 60 when they're diagnosed with this leukemia. If you look at the survival from when I trained in the 80s until today, it is about 20% or less if you're over 60. And if you look at the time course, it has not changed hardly at all in 4 decades. So an extremely common leukemia and an incredibly lethal leukemia. So much more complicated disease. And if you look at drug development, we haven't approved hardly any drugs in the past 3 decades. And part of the reason we haven't approved drugs in the past 3 decades is that none of the precepts that I talked about for targeted therapies have been applied to AML. It's an incredibly heterogeneous disease, and most drug development is let's treat all comers with AML, not recognizing that there are lots of different subtypes. So why would we expect one drug to work when a disease is incredibly heterogeneous? There is some genotype-specific therapy for a relatively rare subtype called acute promyelocytic leukemia, which demonstrates that if you are genotype-specific, you can have successful therapies. But then active drugs get tested in the relapsed refractory setting. So again, the precept of treating early in the course of the disease, you're treating relapsed refractory or even more heterogeneous and resistant, and you expect enormous activity. Again, not very likely to happen. So despite all that, we actually know more about AML than virtually every other cancer on the planet. So it's just remarkable to me that we haven't done anything. So when you look at AML, this is Tim Lay's work with the TCGA, there's a relatively small number of somatic mutations in AML, and here they're listed from FLT3, DMT3A, NPM1. Again, we know all the major drivers, all the molecular abnormalities. But the problem is this is the work from Ross Levine at Sloan Kettering. On the left is the list of all the molecular drivers, and on the right is how they can all combine. And so now, instead of 20 different subtypes, there may be 100 different subtypes that has a FLT3 and an NPM1, or a DMT3A and an IDH2. So now we start to see how heterogeneous this disease really is. But when you sit down and think about it, there are lots and lots of targets. And I'm not going to go through all the targets on the slide there. They were just listed there. But the reality is, for most of these subtypes, we actually have drugs that could be deployed. IDH1, IDH2, FLT3, we have a lot of the drugs in our armamentarium, but we're not deploying them in a rational manner. So, a couple of years ago, I was at a meeting convened by the Leukemia and Lymphoma Society, and a group of the leukemia AML experts got together, and everybody talked about why we couldn't do a master protocol. It was, well, you can't turn around the genomics fast enough. Well, you won't get enough patients. And it was all this skepticism. And I've lived through some pretty skeptical days, and I was discussing with Eric earlier, kinase inhibitor for CML, you're going to shut down every kinase in the kinome. It'll never work. You'll never see a return on investment. So again, I've lived through skeptics. And after the meeting, I sat down with one of the people at the Leukemia and Lymphoma Society and said, there are a couple people in the room that I think that actually believe in it. John Bird from Ohio State, Ross Levine from Sloan Kettering. Why don't the three of us get together with your backing, and we'll put together this master protocol, biomarker-based. And for the next 2 years, we were on weekly phone calls, meeting with the FDA, meeting with drug companies, and we ultimately launched a master-based protocol. And Laura, I'm sure you know what I spy. This is really, really hard work. So what's our trial? We want to perform genomic screening within 7 days of clinical trial enrollment. So think about that. Patient enrollment, turnaround genomics in 7 days. That hasn't been done. And we're working mostly with Foundation Medicine, and we really had to push them to get these turned around in 7 days so we could go from a patient to enrollment. We want to assign therapy based on the genomic screening. We want to have a marker-negative arm, so if patients don't have any of the markers, they can still get on a treatment trial, maybe targeting a cell surface marker. So again, the idea is we want to get everybody funneled into a clinical trial. Each arm is independent with consistent eligibility. So the eligibility, you're over 60, not eligible for chemotherapy, you can enroll in this trial. We'll screen you, we'll assign you to a treatment. It's a window design in untreated patients. So we treat early, not relapsed refractory. We're treating as early as we can before the disease evolves and develops all sorts of other abnormalities. And we work with the FDA. If we see large effects, this could be a path to drug approval. So the design, patient signs up, they get their marrow, we send it to a central genomic screening provider, they turn it around in 7 days, they're assigned to treatment, and they get started on the trial. So again, pretty simple master protocol with sub-designs. And again, I'm not going to walk through all of this. We currently have 2 arms open, a CD33 and a CD200 antibody. This month, we'll open a SYK inhibitor and IDH2. By April, we'll have a FLT3 and an IDH1 inhibitor, and by June or July, we'll have 2 additional arms. So 9 treatment arms on this protocol by June of this year. We've already opened with the 2 arms I've noted. So the trial opened at 2 sites, my site in Oregon and John Bird's at Ohio State in late November. And at 2 sites, we've enrolled 20 patients in 2.5 months. Sloan Kettering's coming on this week, and 5 or 6 others are joining us within the next couple of months. All but 1 of those patients had their genomics turned around in 7 days. Absolutely remarkable. The one who failed, there was a quality control issue, and it was over the holidays. So I think we're doing pretty well. If we can keep on a 95% 7-day turnaround, that would be pretty remarkable. Only 9 of 20 have enrolled on the treatment trials, and that has to do with the fact that we only have 2 arms open, and one of the arms had to shut down temporarily because it was partial dose escalation. But even so, we're running close to 50% of the patients that sign up get on the treatment. There are also a couple of screen failures. They didn't actually have AML. So we're actually doing pretty well on this, and we're outpacing the expectations for trial accrual. And as we bring more sites on, our view is with 10 arms, 20 sites, we can accrue this very, very quickly. So right now, as I mentioned, it's open at Ohio State and OHSU. We're bringing on Sloan Kettering and Dana-Farber Mass General will come on. 6 more sites by April 2017, and by the end of this year, we'll expand to about 20 sites. But as with any effort like this, it's all about collaborations. This has been Leukemia and Lymphoma Society being the neutral broker for all of us. It's been patients who've been advocates for this. It's been drug companies putting their most promising agents into the trial. It's been an advocacy agency like ASH actually driving patients to the trial. A clinical research organization to help us run a master protocol across 20 sites. It's been 11 academic centers that actually agree that they're going to put this through a central IRB, that they're going to use a central clinical research organization, and a genomic provider who stepped up and said, yes, we can do this and we'll help you. And managing all that, and that has been really, really difficult. But again, if you put patients first, they want this, they need this, and if we're going to make an impact, this is the kind of collaboration that we need. And so I'm really proud of this effort, and I have to say that it's been John Bird and Ross Levine. I've been the cheerleader. They've been doing all the work, and I get on the calls, and they just have get this done. And then Amy Bird at Leukemia and Lymphoma Society has been the taskmaster, keeping us all moving forward and getting it done. So we have this master trial that's open. We have genomics being performed in 7 days. We really want this to be a trial where if you get screened, you get put on a treatment arm. It's a master protocol with uniform eligibility, and we have a lot of flexibility to move arms in, move arms out. We're looking for big effects. We want to see much improved therapy for patients with this leukemia. And we also have built in a phase 1B portion so we can do novel combinations, novel combinations with 2 companies providing their drugs. We want those opportunities. So that's great, but I want to spend just the last couple of minutes talking about, are there lessons from the past that we can now provide to precision medicine of the future? And having lived through an era where we've seen remarkable advances, I just want to share some of my own thoughts about this. And what I want to do is I want to take you back to 1900. Leading causes of death in our country: infectious diseases, pneumonia, tuberculosis, diarrheal diseases. You don't even see cancer on the top 5 list. It's just not there. People didn't live long enough because life expectancy was 47 years. So you fast forward to 2000. Heart disease number 1, sorry, Eric. Cancer number 2, but we're going to catch up. And perhaps with the obesity epidemic, it'll stay like this. But nonetheless, if you think about what happened over this century, and I've had this slide in my deck a long, long time, at least since 2000, but I've kept it at 1900 because a lot happened over this last century, starting in 1900. Things like sanitation and refrigeration. We didn't have sewage treatment facilities until 1903. We didn't pasteurize milk until 1906, 1907. Refrigeration wasn't even common until World War II, 1940s. So think about keeping our food and water supply safe. That wasn't even done before 1900s. If you talk to somebody about targeted therapy for bacterial infections in 1900, they would have thought you were from another planet. And penicillin was discovered in 1928 and wasn't through until the war effort in the 1940s. Sulfas were just a little bit earlier than that. Vaccinations, 1950s. But let me recast that. That essentially is prevention, specific treatments, and immune modulation. And if we think about what we're going to do for cancer in the 21st century, it's going to be specific targeted therapies like I've been talking about, like Imatinib, like some of the other targeted therapies with precision medicine. It's going to be prevention and early diagnosis, but also in a much more precise, targeted manner based on an understanding of disease pathogenesis and immune modulation. And again, it's not going to be, let's just throw immune therapies at everybody. It's going to be who needs what therapy, how is it going to work, is it going to be more targeted, and can we identify patients that it will work for or it won't work and why it won't work. And so what I'm really saying is that if we're going to make advances, we need to understand what we're doing. And if we understand what we're doing, we can see remarkable advances. So you're probably thinking, well, okay, that's interesting. How can Dr. Druker Brian make that apply to me? So I'll have some help here, and you can see how it can apply to you.
Everybody, come here. Gentlemen, it would seem that the germ of syphilis has been discovered. Really? By whom? By one Fritz Schaudin. The German Medical Weekly sent me these proofs for approval. Listen. The spirocheta pallida is a protozoa. It is a fine, steeply convoluted filament with 6 to 14 turns. It is decidedly motile, with forward, turning, and bending movements. Tell me, does that description put you in mind of anything else? It is decidedly motile, with forward, turning, and bending movements. Why, that's just like the trypanosome. Right. If the germ has been discovered, there's cause to hope for a cure. Yes, there is hope.
Thank you, Hollywood. If the cause has been discovered, there's hope for a cure. And ultimately, that's our job for the 21st century, is to make that hope a reality for all of our patients. And these are some of my phase 1 Gleevec patients, many of them still alive and thriving because of what we were able to accomplish for them. And again, this is what I want to leave you with, is that hope and that promise that we can make an impact against these diseases by understanding them and applying genomic medicine principles. Thank you very much.